How could a fungus learn to consume plastic? How has nature begun to fight back against the plastic nightmare created by humanity? And most importantly, what if I told you this fungus could prevent a future catastrophe? Somewhere deep in the Amazon rainforest, tucked beneath the canopy of one of the most biodiverse ecosystems on the planet, a student crawling through the undergrowth on her hands and knees made a discovery so strange, so improbable, that it would quietly ripple through the scientific community for years to come. Actually, she wasn't looking for what she found, but there it was, a small, unassuming mushroom growing directly out of a piece of discarded plastic, quietly consuming it, breaking it down, turning one of humanity's most stubborn and catastrophic creations back into something the Earth could recognize. That student was Priya Anand, a Yale undergraduate on a research expedition in Ecuador in 2011. And the mushroom? It was Pestalotiopsis microspora, a fungus previously known only as a mild plant pathogen, living its quiet life on tree bark and decaying wood in tropical forests. Nobody had asked it to evolve this extraordinary capability. Nobody had engineered it in a lab. It had simply done what fungi have always done throughout the nearly billion-year history of their kind. It had found a way. And that, right there, is the beginning of one of the most remarkable and underreported stories in the history of biology. A story that asks us to completely rethink our relationship with the organisms we most frequently overlook, underestimate, and frankly just find a little bit gross. A story that could, if we're willing to listen carefully enough, offer us a genuine path out of one of the defining environmental crises of our age. But before we delve into this topic, before we discuss what this means for the mountains of plastic waste that are currently choking our oceans, filling our landfills, and even seeping into our bloodstreams, we need to understand what we're actually dealing with. We need to understand fungi. We need to understand them not as they're described in a diagram in your high school biology textbook, squeezed in between plants and animals as if to apologize, but as a forager understands the forest floor, or as a mycologist feels when, kept awake by what they've seen, they peer through a microscope at 3 in the morning. We need to know just how ancient, how capable, and how strange these organisms really are. Because here's the point, a fungus learning to eat plastic is the latest expression of billions of years of adaptive genius that has survived ice ages, mass extinctions, continental collisions, and the slow drowning of oceans. Fungi were solving impossible problems long before we came to create them. And now, against all odds, they may be poised to solve our problems. Let's get something out of the way right from the start. If you're a mushroom enthusiast, and if you're here, you probably are, you already know that fungi get a raw deal in popular science. They've spent most of recorded history being misidentified, misunderstood, or just plain ignored. For centuries, botanists argued about whether they were even alive in any meaningful sense. The great Carl Linnaeus, who gave us the biological classification system we still use today, essentially threw his hands up when it came to fungi. He called them chaos. Literally, that was his scientific term. He could not figure out what to do with them, and so he named them for disorder itself. It didn't help that fungi refuse to behave like anything else we're familiar with. They're not plants, even though they stay in one place and grow from the ground. They don't photosynthesize. They don't have leaves, roots, or seeds in any conventional sense. They're not animals either, even though their cellular chemistry is often more similar to ours than to any plant you could name. They occupy this strange and deeply uncomfortable middle ground, and for a scientific tradition that has always loved neat categories, that made them difficult to love. It wasn't until 1969 that fungi were finally granted their own kingdom, a scientific acknowledgement long overdue that they represented something genuinely distinct from everything else on Earth. And even then, for much of the 20th century, they remained the poor cousins of the biological world. Botany had its prestige, zoology had its charisma. Mycology, the study of fungi, was a discipline practiced by enthusiasts who were widely regarded as slightly eccentric, perhaps a little too comfortable with the smell of decay. But here's what those enthusiasts knew that nobody else did. Here's what was becoming increasingly obvious to anyone who actually spent time paying attention to the fungal kingdom. These organisms are not peripheral. They are not background noise in the symphony of life on Earth. They are, in many ways, the foundation on which that symphony is built. Without fungi, most of what we recognize as a healthy terrestrial ecosystem would simply cease to function. The trees in your local forest would be unable to absorb sufficient nutrients from the soil. Dead leaves, fallen logs, and the bodies of animals would pile up undecomposed, locking away nutrients that the living world desperately needs. The soil itself, that complex, dark living matrix that makes agriculture possible, would be a pale and impoverished shadow of what it currently is. Fungi are the great decomposers, the great recyclers, the great connectors. They are the invisible infrastructure of the living world. And they have been doing this work quietly and without recognition for somewhere between 800 million and 1 billion years, possibly longer. We are genuinely not sure how long they have existed, because the deeper paleontologists dig, both literally and figuratively, the further back the fungal story seems to stretch. Would you like to add anything, Chloe?
Yes, of course. Actually, the only thing we know for sure is this. When the first plants began their extremely slow transition from aquatic to terrestrial life, about 450 to 500 million years ago, fungi were already there, already established on the land surface, already breaking down rock, creating soil, building the biochemical infrastructure that those first pioneering plants would need to survive. The two kingdoms, plants and fungi, almost certainly formed one of the most important alliances in the history of life. Plants provided sugars, manufactured through photosynthesis. Fungi provided minerals and water, extracted from rock and soil through the extraordinary chemical power of their hyphal networks. It was a deal struck in the Ordovician period, and many of the trees in your backyard are still honoring it today. The roots of most land plants are not independent structures. They are colonized by fungal networks, interwoven with hyphal threads, in what mycologists call mycorrhizal associations, from the Greek for fungus and root. The plant and the fungus are not separate entities operating in proximity. They are a partnership so ancient and so integrated that separating them in any meaningful biological sense is almost impossible.
So, when we talk about a fungus that can eat plastic, we are not talking about a curiosity, an oddity, a footnote. We are talking about the latest trick from an organism with 1 billion years of experience in breaking down materials that nothing else can touch. We are talking about a kingdom that has survived everything the Earth has thrown at it, including the conditions of space, as experiments on the International Space Station have confirmed, and which is now, apparently, turning its attention to the particular kind of mess we've made. And that mess, as you almost certainly know, is very, very large.
Yeah, absolutely. It's difficult to overstate how thoroughly plastic has saturated the modern world. We live inside it, eat food packaged by it, drink water from bottles made of it, wear clothes woven from synthetic fibers derived from it. Our hospitals depend on it. Our technology is built around it. The device you're likely using to watch or listen to this contains dozens of different plastic components, some of them thinner than a human hair. Plastic, in the half-century or so since it became ubiquitous, has become so deeply embedded in the fabric of modern life that imagining its absence requires a genuine effort of imagination. And yet, plastic is, in the most fundamental sense, a material that the natural world has no idea what to do with. Let's back up and think about what plastic actually is. Most conventional plastics are derived from petroleum, from the same ancient compressed hydrocarbons that we burn in our cars and power plants. They are essentially chains of carbon-based molecules called polymers, assembled through industrial processes into materials with specific properties: rigidity, flexibility, transparency, conductivity, insulation. The specific arrangement and length of these polymer chains determines what kind of plastic you end up with. Polyethylene terephthalate, or PET, is the clear plastic in your water bottle. High-density polyethylene, or HDPE, makes your shampoo bottle and your grocery bag. Polyurethane makes foam cushioning. Polystyrene makes the foam cup your coffee shop still occasionally uses despite knowing better.
These are remarkable materials in many ways. They are cheap to produce, durable, lightweight, and incredibly versatile. The problem is that their durability does not switch off when you throw them away. A plastic water bottle tossed into the trash in 2024 will still be recognizably plastic in 2424. Most estimates suggest that conventional plastics take between 450 and 1,000 years to decompose under normal environmental conditions, and even then, they don't really decompose in the way that an apple core or a wooden log decomposes. They don't break down into constituent elements that the biological world can readily reabsorb. They fragment, they shatter into smaller and smaller pieces, down to the microscopic scale, until they become what we now call microplastics, particles smaller than 5 millimeters, often far smaller, which are now found quite literally everywhere on Earth. In the deepest ocean trenches, in Arctic sea ice, in the tissues of fish, birds, and marine mammals, in the flesh of the fish you might have had for dinner last night, in the water coming out of your tap, and yes, in your blood, in your lungs, in the placentas of unborn children. A landmark study published in 2022 found microplastics in human blood samples for the first time, with traces detectable in nearly 80% of the people tested. We do not yet fully understand what this means for human health, but the early indications are not reassuring. Microplastics have been associated with inflammation, endocrine disruption, and damage to cellular membranes. They carry with them the chemical additives, plasticizers, flame retardants, colorants that were incorporated into the original plastic and which tend to be toxic in their own right.
The numbers involved are genuinely staggering. Since the large-scale production of plastic began in the early 1950s, humanity has produced somewhere in the region of 9.1 billion metric tons of the stuff. To put that in perspective, that is more than the combined weight of every human being who has ever lived. Of that total, only about 9% has been recycled. Another 12% has been incinerated, which creates its own set of toxic byproducts. The remaining 79% has ended up in landfills or in the natural environment, in our oceans, our rivers, our soils, our air. Current production rates show no sign of meaningful decline. If anything, they are accelerating. Global plastic production is expected to triple by 2050 relative to 2014 levels, even as international negotiations over plastic pollution continue with painful slowness. The oceans currently contain an estimated 150 million metric tons of plastic, with 8 million metric tons added every single year. And because plastic does not degrade in any meaningful time frame, every piece that has ever entered the ocean is, in some form, still there. It is, by any reasonable measure, one of the most significant environmental crises of the modern era. It is ubiquitous, it is growing, and until very recently, nobody had any particularly convincing idea of what to do about it. Reduction is necessary but politically fraught. Recycling is inadequate, and increasingly revealed as having been somewhat oversold as a solution. Cleanup efforts, however heroic, are struggling against a flow that vastly exceeds their capacity. The math simply does not work in our favor. And then, a student in Ecuador found a mushroom growing out of a piece of polyurethane foam.
Let's go back to that rainforest. Let's spend a little time understanding what Priya Anand actually found and why the scientific community's response to her discovery was so significant. The Yale student research program that took Anand to Ecuador in 2011 was part of an ongoing effort to catalogue the extraordinary biodiversity of the Amazon basin, a project of obvious urgency given the pace at which that biodiversity is being lost. Students were collecting samples of fungi from plant tissues throughout the forest, the idea being to screen these so-called endophytic fungi that live inside plant tissues without causing disease, for potentially useful biological compounds. It is the kind of broad-net exploratory science that occasionally turns up something genuinely transformative. This was one of those occasions. When the team returned to Yale and began testing their fungal isolates, they discovered something that stopped them cold. Several of the samples they had collected were capable of growing on polyurethane as their sole carbon source, not growing alongside polyurethane or growing on a substrate contaminated with polyurethane, growing on polyurethane itself, using it as food, breaking it down enzymatically and incorporating the resulting molecular fragments into their own biological processes. The most potent of these plastic-eating isolates was Pestalotiopsis microspora, and what made it particularly interesting and genuinely extraordinary was that it could do this not only in the presence of oxygen, but in its complete absence, anaerobically. In conditions mimicking the oxygen-depleted interior of a landfill, where most biological degradation processes grind to a halt entirely, Pestalotiopsis microspora just kept eating. The key to this ability, as subsequent research would reveal, was a specific enzyme. Enzymes are the molecular workhorses of biological chemistry, proteins that catalyze specific chemical reactions, breaking or forming particular bonds with extraordinary precision and efficiency. The enzyme in question, which the researchers called a serine hydrolase, was capable of attacking and breaking the ester bonds that hold polyurethane together. It could, in essence, read the molecular structure of polyurethane and find the weak points, the chemical joints, and pull them apart. This was not a trivial finding. Polyurethane is one of the most widely used plastics in the world, found in everything from foam mattresses and insulation panels to car dashboards, medical devices, and the soles of your shoes. It is estimated that global production of polyurethane exceeds 18 million metric tons per year, and it is notoriously difficult to recycle. The discovery that a fungus could enzymatically degrade it, even anaerobically, was genuinely news.
But here is where the story becomes even more interesting. Because Pestalotiopsis microspora was not an isolated discovery. It was, in retrospect, the opening of a door that revealed a much larger room than anyone had anticipated. Once researchers knew what to look for, once the possibility had been established that fungi could evolve the capability to degrade synthetic polymers, similar discoveries began to accumulate with remarkable speed. In 2017, researchers reported that Aspergillus tubangensis, a fungus previously known mainly as a spoilage mold on food and a producer of various useful enzymes, was capable of degrading polyurethane through a combination of enzymatic activity and the physical action of its growing mycelium. The hyphal threads of this fungus probing into the plastic surface, literally broke apart the polymer chains through mechanical force, while simultaneously secreting enzymes that continued the chemical degradation. It was a 1-2 punch, physical and chemical degradation, working in concert, that proved more effective than either mechanism alone. In 2016, in perhaps the most widely publicized finding in this rapidly expanding field, Japanese researchers announced the discovery of a bacterium, Ideonella sakaiensis, capable of degrading PET plastic, the material in your water bottles, with astonishing efficiency. The organism had apparently evolved this capability in a PET-rich environment outside a plastic recycling facility, a piece of real-world evolution playing out on a timescale of decades rather than millennia. The bacterium produced two novel enzymes, PET-ACE and MHET-ACE, which worked sequentially to break PET down into its constituent monomers. And while Ideonella sakaiensis is a bacterium rather than a fungus, the discovery reinforced the broader point that microbial life, given sufficient exposure to a new carbon source, will eventually find a way to exploit it. Fungi have since been identified in a growing catalogue of plastic-degrading species: Aspergillus niger, Penicillium simplicissimum, Cladosporium cladosporoideodes, Bjerkendera adusta. The list keeps growing, and the variety of plastics being targeted keeps expanding. Polystyrene, long considered one of the most recalcitrant of all polymer types, has been found to be degradable by certain fungi. Polyethylene, the ubiquitous plastic of grocery bags and packaging films, has been attacked by fungal isolates capable of using the long hydrocarbon chains as a carbon source, albeit slowly. Even PVC, that most chemically resistant of all common plastics, has been shown to be susceptible to fungal modification under certain conditions. And then there was Chernobyl.
We need to talk about Chernobyl for a moment. Not because it's directly about plastic, but because it tells us something profound about the nature of fungal adaptation that is absolutely essential for understanding why plastic degradation is not just plausible, but almost inevitable. When reactor number 4 of the Chernobyl nuclear power plant exploded on April 26, 1986, it released a cloud of radioactive material 20 times more intense than the combined fallout of the atomic bombings of Hiroshima and Nagasaki. The immediate area was devastated. Within weeks, the forest surrounding the plant, known now as the Red Forest, had turned the color of rust from radiation damage, the trees killed outright by the overwhelming bombardment. Animals that couldn't flee perished. The soil and water was saturated with cesium-137, strontium-90, and plutonium isotopes that would persist for decades, centuries, in some cases thousands of years. And yet, into this extraordinary wasteland, fungi crept back almost immediately.
Not just surviving the radiation, mind you, using it. Researchers studying the fungi that colonized the abandoned plant, including the intensely radioactive water still cooling the remains of the reactor core, found something that shouldn't have been possible by any conventional biological logic. Certain fungi, notably species of Cladosporium, Cryptococcus, and Wangia, appeared to be using the ionizing radiation as an energy source, in much the same way that plants use the electromagnetic radiation of sunlight. The fungal cells were packed with melanin, the same pigment that darkens our own skin in response to ultraviolet radiation, and which in these fungi appeared to be acting as a kind of radiation harvesting antenna, absorbing the energy of radioactive decay and channeling it into cellular metabolism.
This process, called radiosynthesis, was first described in 2007, and it remains one of the most mind-bending discoveries in modern mycology. The implications are profound on multiple levels. At the most fundamental level, it suggests that the energy sources available to biological life are broader than we had imagined. Photosynthesis and chemosynthesis have long been known as the two great primary production strategies, the mechanisms by which organisms capture raw energy from the environment and use it to drive biological processes. Radiosynthesis appears to be a third, previously unknown option, and it is fungi that appear to have discovered it, here in the ruins of our worst nuclear accident. But for our purposes, what matters most about the Chernobyl fungi is not the specific mechanism, it is what it tells us about fungal adaptability. These organisms, encountering a form of environmental stress that has no natural precedent in their evolutionary history, no mushroom ever evolved under the shadow of a nuclear reactor, responded not by dying, but by finding a way to turn the stressor into a resource. They took something deadly and made it into food.
This is what fungi do. This is what they have always done. It is the deepest expression of the hyphal intelligence, if we can call it that mycologists have been attempting to characterize and describe for decades. The probing, branching, fusing networks of fungal hyphae are not passive structures. They are active explorers, constantly sampling their environment, constantly testing new substrates, constantly evaluating the energetic cost and benefit of pursuing or abandoning any given chemical trail. And when they encounter a new material, even one as alien as synthetic polymer or ionizing radiation, their evolutionary toolkit, refined over nearly 1 billion years of continuous adaptation, gives them a remarkable capacity to eventually find a way in. The plastic-degrading fungi are not, from this perspective, surprising at all. They are doing exactly what fungi do. The surprise is only that it took us so long to notice.
Let's get into the actual science here, because this is genuinely fascinating stuff, and you deserve the full picture. How exactly does a fungus break down plastic? What's happening at the molecular level when Pestalotiopsis microspora sinks its enzymatic teeth into a piece of polyurethane foam? To understand this, we need to briefly talk about how plastic is structured and why conventional biological degradation processes struggle with it. Most synthetic polymers are essentially very long chains of repeating molecular units, the monomers, linked together by specific types of chemical bonds. The kind of bond matters enormously. Biological degradation generally works by deploying enzymes that recognize and break specific bond types. The reason that most plastics resist biological degradation is that their specific bond types, ester bonds in PET and polyurethane, and carbon-carbon bonds in polyethylene and polystyrene, are either absent from or extremely rare in the natural biological environment. Enzymes evolve to break the bonds that naturally abundant substrates present, and for most of life's history, synthetic polymers simply weren't around to present themselves as substrates.
But fungi, particularly in the white rot and brown rot categories that live by decomposing wood, have spent millions of years evolving enzymes capable of attacking some of the most chemically resistant biological polymers in existence. Lignin, the compound that gives wood its structural rigidity, is one of the most recalcitrant natural polymers in biology. Breaking it down requires an enzymatic arsenal of remarkable power and versatility, including a family of enzymes called laccases and peroxidases that work by generating highly reactive chemical species called free radicals, which then attack the polymer in a relatively non-specific way, breaking bonds across a wide chemical range.
Exactly. It is precisely this chemical versatility, this ability to generate broad-spectrum reactive species rather than targeting a single specific bond type, that appears to give certain fungi a head start when it comes to synthetic polymers. The lacase enzymes of white rot fungi, like Trametes versicolor, the turkey tail mushroom familiar to many of you as a popular medicinal species, are capable of oxidizing a remarkably wide range of chemical compounds. And when confronted with synthetic polymers, these broadly reactive enzymes appear capable of initiating at least the first steps of degradation, introducing chemical modifications that make the polymer chains more accessible for further enzymatic attack. In Pestalotiopsis microspora, the specific enzyme responsible for polyurethane degradation is a serine hydrolase, a type of enzyme that uses a specific amino acid, serine, as its catalytic center to attack ester bonds. Polyurethane contains urethane linkages, which include ester-type bonds, and the serine hydrolase of Pestalotiopsis microspora appears to be particularly well configured to recognize and attack these specific bonds. The enzyme was likely originally evolved for a different purpose, perhaps attacking the surface coatings of certain plant tissues, and then, over time, in an environment increasingly contaminated with polyurethane waste, underwent the gradual mutational refinements that tuned it towards its new substrate.
This process of enzymatic evolution, the gradual refinement of an existing enzyme's specificity toward a new substrate, is one of the most important and rapid forms of evolutionary change available to microorganisms. Unlike the slow, multi-generational process of morphological evolution, enzymatic evolution can happen very quickly in microbial populations with short generation times and large population sizes. E. coli, to give a well-studied example, can evolve the ability to metabolize a new carbon source in a matter of weeks under selective pressure. Fungi tend to have longer generation times, but their vast genetic diversity and extraordinary metabolic flexibility give them a similarly powerful capacity for rapid enzymatic adaptation. And importantly, fungi have a mechanism for sharing useful enzymatic innovations that goes beyond simple inheritance. They can engage in horizontal gene transfer, the exchange of genetic material between individuals and even between species, that allows a successful adaptation in one fungal organism to spread relatively rapidly through a broader community. The enzyme that one fungus evolves to break down polyurethane may not stay exclusive to that species for long. In the complex microbial communities of soil and forest litter, where fungi are in constant interaction with bacteria and other microorganisms, useful genetic innovations have a tendency to spread. This is, when you think about it, an extraordinary thing. It means that the discovery of a plastic-degrading enzyme in Pestalotiopsis microspora in Ecuador is not merely interesting for its own sake. It is a signal, a glimpse of an evolutionary process already underway, already spreading, already ramping up in response to the massive selective pressure that 7 decades of plastic pollution have applied to the microbial world. We are, in a very real sense, selecting for plastic-eating fungi. The more plastic we put into the environment, the stronger the selective advantage for any organism that can use it as a resource, and the more rapidly those organisms will evolve, diversify, and spread. Nature, it turns out, abhors a wasted carbon source just as much as it abhors a vacuum.
Before we get into what scientists are actually doing with plastic-eating fungi and what the future of this research might look like, it's worth spending some time on something that is absolutely central to understanding why fungal solutions to plastic pollution are so different in kind from the technological solutions we've been pursuing. Fungi, as any serious mushroom enthusiast will know, are not isolated organisms. They are networked. They exist within and in large part constitute an extraordinarily complex web of biological relationships that spans entire ecosystems. The mycorrhizal networks of forest soils, sometimes poetically called the wood wide web, connect individual trees through fungal threads that can extend for kilometers through the soil. These networks don't just passively link trees, they actively facilitate the transfer of nutrients, water, and even chemical signals between connected plants. Carbon, fixed by a tree with abundant sun exposure, can be moved through the fungal network to a shaded seedling that needs it. Warning signals from a tree under insect attack can be transmitted to neighboring trees, allowing them to mount preemptive chemical defenses. This networked existence is not incidental to what fungi are, it is fundamental. The mycelial network is the organism, in a very real sense. The mushrooms you see above ground, the fruiting bodies, the reproductive structures, are just the momentary expressions of a much larger, much older entity spread through the soil below. And that entity is perpetually active, perpetually exploring, perpetually integrating information from across its entire extent. Now think about what this means for plastic degradation. When a fungal mycelium encounters a piece of plastic in the soil, it doesn't just send one hyphal tip to investigate, it sends thousands, millions potentially, each tip probing the surface, each tip reporting back, in whatever sense we choose to understand that term, on what it has found. The enzymatic response is not mounted by a single cell, but coordinated across the entire network. And if one part of the network makes progress, begins to break down the polymer, begins to extract usable carbon resources, from elsewhere in the network can be redirected to support and amplify that success.
This is an inherently scalable process. A fungal solution to plastic pollution is not just a point solution, a single organism attacking a single piece of plastic in isolation. It is, potentially, a network solution, capable of spreading through a contaminated environment the way a mycelial front spreads through forest soil, slowly but inexorably, covering ground that no point source technology could efficiently address. This is an important distinction, because one of the fundamental challenges of plastic pollution is its spatial dispersal. Plastic is not conveniently concentrated in one place where we can send cleanup machinery. It is everywhere, mixed into soils across agricultural land, dispersed through ocean gyres, embedded in river sediments, incorporated into the bodies of animals and plants. Technological solutions to this dispersed contamination face enormous practical challenges. How do you deploy a machine to address a problem that exists simultaneously in a Pacific garbage patch, an Arctic ice core, and the stomach of a seabird on a remote island? Fungi already know how to address dispersed, diffuse substrates in complex environments. They have been doing it for hundreds of millions of years. Their hyphal networks are specifically adapted for penetrating irregular, heterogeneous matrices, soil, wood, leaf litter, decaying animal tissue, and extracting resources from materials that are not conveniently packaged. A mycelial network advancing through contaminated soil is, in principle, exactly the right tool for exactly this problem. So if plastic-eating fungi are real, and they are, and if their networked existence makes them theoretically well-suited to addressing the dispersal problem, and it does, why aren't we already using them at scale? Why are our landfills not full of carefully selected fungal cultures quietly working their way through decades of accumulated plastic waste?
The honest answer is that we're working on it, and the progress, while not always front-page news, has been more substantial than you might expect. The practical challenges of deploying fungi for plastic bioremediation fall into several categories, each of which is the subject of active research. The speed problem. Fungal plastic degradation in its natural form is slow. Pestalotiopsis microspora can break down polyurethane, yes, but it does so over weeks and months under optimal laboratory conditions. In the field, where temperatures, moisture levels, and nutrient availability are less controlled, the process is likely to be slower still. Degrading the billions of tons of plastic already in the environment at this rate would take geological timescales, which is admittedly still faster than leaving it to degrade on its own, but not exactly the urgent intervention we need. Researchers are attacking this problem from several angles. One approach is enzyme optimization using genetic engineering and directed evolution techniques to improve the efficiency of the plastic-degrading enzymes themselves. The PETase enzyme from Ideonella sacchaeus has already been significantly improved in this way. In 2018, a team at the University of Portsmouth and the National Renewable Energy Laboratory in the United States accidentally created a mutant version of the enzyme that was considerably more effective than the natural version. By examining the crystal structure of the enzyme and making targeted modifications to the amino acids around its active site, they produced a variant that broke down PET plastic about 20% faster. Subsequent work has produced even greater improvements, with some engineered enzyme variants breaking down PET in hours rather than weeks. Similar approaches are being applied to fungal enzymes responsible for degrading polyurethane, polystyrene, and other polymer types. The goal is to create a toolkit of highly optimized enzymes that can be produced at industrial scale, either by engineering the original fungal producers or by transferring the relevant genes into faster-growing production organisms like yeast or bacteria. The selectivity problem. A second challenge is ensuring that plastic-degrading fungi or their enzymes degrade plastic and not everything else in the surrounding environment. The enzymatic machinery that breaks down synthetic polymers often has broad activity against natural polymers as well, and releasing highly active fungal cultures into the open environment risks degrading materials we actually want to keep: wood, textiles, building materials, and, crucially, the natural organic matter in soil that supports plant growth and ecosystem function.
This is a genuine concern, and it has prompted considerable caution among researchers who might otherwise be tempted to simply deploy plastic-eating fungi broadly. Current research is focused on developing contained bioremediation systems, essentially industrial-scale composting setups, in which plastic waste can be exposed to optimized fungal cultures under controlled conditions, with the degradation products captured and the fungal communities carefully managed to prevent unintended spread. The product problem. A third challenge is understanding what the fungi actually produce when they break down plastic. Ideally, the end products of plastic degradation would be simple, biologically familiar compounds: carbon dioxide, water, small organic acids that the environment can easily absorb. In practice, the picture is more complicated. Plastic degradation can produce a variety of intermediate compounds, some of which may themselves be toxic or persistent. The additives incorporated into plastic, plasticizers, flame retardants, UV stabilizers, do not disappear when the polymer chains are broken. They must be degraded separately, and not all fungi are equally capable of handling all additive types. Research into the full metabolic pathways of plastic degradation, tracking every molecule from the initial polymer through all intermediate stages to the final products, is ongoing and will be essential for assessing the safety and environmental impact of large-scale bioremediation. The goal is not merely to break plastic down, but to mineralize it completely,
to convert it entirely into inorganic compounds that represent no ongoing environmental risk. The scale problem. And then there is the sheer scale of the challenge. We have produced 9.1 billion metric tons of plastic. Even an extremely efficient bioremediation process would take enormous time and physical resources to address that backlog. The most realistic near-term applications for plastic-eating fungi are probably not in the open environment at all, but in industrial settings, at waste management facilities, at recycling operations, at production sites where plastic waste streams are concentrated and predictable. Several companies are already moving in this direction. Novozymes, a Danish biotechnology company and one of the world's largest producers of industrial enzymes, has been working on enzyme-based PET degradation. A French company called Carbios has developed an industrial process using an engineered version of a bacterial PET degrading enzyme that can break down a metric ton of plastic in under 24 hours, producing the original monomers in high enough purity to be repolymerized into new plastic without quality loss, a genuine chemical recycling process that could, if deployed at scale, close the loop on PET waste. Their first industrial-scale demonstration plant is already operational in France. The fungi are not yet the stars of these industrial applications, the bacterial enzymes, being more thoroughly characterized and more amenable to industrial production, have moved somewhat faster. But fungal contributions are increasingly present in the research pipeline, particularly for polymer types that bacteria have proven less capable of addressing. Polyurethane, polystyrene, and the mixed polymer composites that make up so much modern electronic and automotive waste are emerging as fungal specialties.
Here's something that might surprise you and might also make you feel rather differently about some of the mushrooms you may already know and love. Several of the most promising fungal candidates for bioremediation are not obscure tropical species found in remote rainforests. They are species that are familiar to mushroom enthusiasts across North America. Species that are widely foraged, cultivated, and consumed. Species that your fellow enthusiasts may already have growing in logs or substrate bags in their backyards.
Tometes versicolor, turkey tail, has already been mentioned for its extraordinarily versatile lacase enzymes, which appear capable of initiating degradation of multiple plastic types. But turkey tail is also one of the most widely distributed fungi in North America, fruiting on dead hardwood from coast to coast. It is robust, adaptable, and easily cultivated. It is already the subject of substantial mycological research for its immunomodulatory properties, which means its biochemistry is among the most thoroughly characterized of any wild fungal species, and its lacase enzymes, which have been studied extensively in the context of bioremediation of dye-contaminated textile wastewater, are now being investigated specifically for plastic degradation.
Pleuretus austriatus, the oyster mushroom, beloved by cultivators everywhere for its speed of growth, ease of cultivation, and excellent culinary qualities, has demonstrated the ability to degrade both petroleal hydrocarbons and certain plastic polymers. Its lignin-degrading enzyme system, powerful and versatile, provides a broad enzymatic toolkit that gives it a head start when confronting novel substrates. Research groups in several countries have demonstrated oyster mushroom degradation of low-density polyethylene, the polymer in plastic bags and food packaging films, under both laboratory and semi-field conditions. The degradation rates are slow. We're talking about meaningful mass loss over months rather than days, but they are real, and they suggest that with optimization, oyster mushrooms or their enzymes
could play a role in addressing the polyethylene waste stream.
Ganoderma lucidum, reishi, the mushroom of immortality in traditional East Asian medicine, has been shown to produce enzymes capable of attacking several plastic types, including polyurethane. Like turkey tail, reishi is extensively cultivated, and its biochemistry is relatively well characterized, making it a tractable target for enzyme optimization and industrial application.
Aspergillus tubangensis, the species reported in 2017 to degrade polyurethane, is less glamorous. It's a mold rather than a fleshy mushroom, not something you'd want to eat, but it represents an important class of fungi with plastic degrading capability. Aspergillus species are among the most widely distributed fungi in the world, found in soil virtually everywhere. The fact that a plastic-degrading capability has been identified in this globally ubiquitous genus suggests that similar capabilities may already be evolving in countless locations around the world, wherever plastic contamination is present. For the mushroom enthusiast, all of this is genuinely exciting. The species you've been growing in your garage or hunting in your local woods aren't just food or medicine, or objects of beauty. They are, potentially, agents of environmental restoration. They carry within their enzymatic arsenals capabilities that, properly understood and deployed, could contribute meaningfully to one of the most pressing environmental challenges of our time. The next time you're watching your oyster mushroom colonize a bag of straw, consider that the same biochemical machinery colonizing that substrate might, with some refinement, be turned toward a bucket of plastic bags. The next time you find turkey tail brackets on a fallen oak, think about the lacase enzymes doing their work on the lignin within that wood and consider what else those enzymes might be capable of. The broader field of mycoremediation, using fungi to remediate contaminated environments, is older than the plastic-specific work we've been discussing, and it provides an important context for understanding both the potential and the limitations of fungal solutions to pollution. Mycoremediation as a formal concept was extensively developed and popularized by Paul Stamets, a Washington state-based mycologist who has spent decades investigating and advocating for the environmental applications of fungal biology. Stamets' 2005 book, Mycelium Running, brought the concept to a wide popular audience, and his TED Talk on the subject remains one of the most viewed science talks in the platform's history. His work has focused particularly on the ability of wood-decomposing fungi to break down petroleum hydrocarbons, the oils, fuels, and industrial solvents that have contaminated soils around the world. The results of early mycoremediation trials with petroleum contamination were genuinely striking. In one frequently cited study, piles of soil contaminated with diesel fuel were inoculated with oyster mushroom spawn and compared to control piles treated with conventional bacterial bioremediation agents. After 4 weeks, the unchanged appearance dark, oily, and presumably still toxic, the oyster mushroom-treated piles were covered in a flush of healthy white mushroom caps, and chemical analysis revealed that the petroleum hydrocarbons had been reduced by more than 95%. Within 8 weeks, attracted by the mushrooms and the now nutrient-rich soil, insects, birds, and plant seedlings were colonizing the formerly barren piles. This last point deserves emphasis. One of the unique features of mycoremediation, as compared to conventional chemical or technological remediation approaches, is its capacity to initiate a cascade of biological recovery. When fungi begin to break down a pollutant in contaminated soil, they don't just remove the pollutant, they begin to restore the biological infrastructure of the soil itself, the complex community of microorganisms, the network of fungal hyphae, the structure of organic matter
that supports plant growth and ecosystem function. Contaminated soil remediated by fungi doesn't just become less toxic, it becomes alive again. The remediation and the restoration happen simultaneously. For plastic contamination in soil, the same cascading recovery is at least theoretically possible, and several pilot studies have begun to explore this. Contaminated agricultural soils inoculated with cocktails of plastic-degrading fungi alongside other mycorrhizal species have shown both increased plastic degradation rates and improvements in soil structure and fertility measures. The fungi are not just eating the plastic, they are, in the process of doing so, rebuilding the soil ecosystem around them. This is a remarkable and underappreciated point. We are not, in the fungal approach, simply exchanging one problem for another or creating a sterile end state. We are potentially initiating a process of genuine ecological restoration, one in which the removal of plastic contamination is part of a larger renewal of soil health, plant community structure, and ultimately ecosystem function. The fungi, as they have been doing for hundreds of millions of years, are preparing the ground for life.
We've spent a lot of time talking about fungi as the solution to the plastic problem we've already created. But there's another angle to this story that is equally exciting and perhaps even more transformative. Fungi as an alternative to plastic itself. Fungi don't just eat plastic. They can also be used to make materials that serve many of the same functions as plastic, but which decompose harmlessly in the natural environment when their useful life is over. The fungal mycelium, that dense network of hyphal threads that we've been discussing throughout this piece, can, under the right conditions, be induced to form a coherent, solid material. The mycelium grows through and around a substrate of agricultural waste, corn stalks, hemp fiber, cotton hulls, binding it together into a rigid, lightweight, heat-resistant structure that can be molded into virtually any shape. The result is a material with genuinely remarkable properties. It is strong, it is lightweight, it is an excellent thermal and acoustic insulator, it is fire-resistant and water-repellent. It is produced without any significant energy input beyond what is needed to maintain the growing mycelium. No high-temperature manufacturing processes, no toxic chemical reagents, no petroleum feedstocks, and, at the end of its useful life, it composts in a matter of weeks. This material, fungal mycelium composite, or what is increasingly being marketed as mycelium packaging, is already on the market. Ecovative Design, a company founded in 2007 by Eben Bayer and Gavin McIntyre in Troy, New York, has been producing mycelium-based packaging commercially for more than a decade. Their products are used by companies including Dell, IKEA, and several prominent wine and spirit brands as a replacement for expanded polystyrene foam. The material performs comparably to EPS in most packaging applications, costs comparably at commercial scale, and generates a fraction of the environmental impact.
Ecovative and companies following in their wake, there are now dozens of mycelium materials startups globally are also exploring applications beyond packaging. Mycelium leather, which has a texture and durability comparable to animal hide, is being developed for the fashion industry by companies like Bolt Threads, whose Milo material has been used by Stella McCartney, Adidas, and Lululemon. Mycelium insulation panels are being trialed in construction. Mycelium composites are being evaluated for automotive interior components. Even mycelium-based medical devices are in development, exploiting the biocompatibility and tunable mechanical properties of fungal biomaterials. And then is what some researchers are beginning to call mycotecture, architecture made from or incorporating living fungal mycelium. The concept is still largely experimental,
but it is advancing rapidly. The idea is not merely to use mycelium composites as building materials, though that alone represents a significant opportunity, but to incorporate living mycelial networks into built structures. Networks that can grow, self-repair, respond to environmental conditions, and potentially even perform sensing and communication functions. Buildings that are, in some meaningful sense, alive. NASA has taken this concept seriously enough to fund research into mycelium-based habitats for space travel and planetary colonization, a development that would surely have astonished those mid-20th century mycologists who couldn't get their colleagues to take fungi seriously as ecological agents, let alone as the basis for the next generation of spacecraft. The possibility space here is genuinely staggering. Fungi that eat our plastic waste, fungi whose bodies replace plastic in the first place, fungal networks that serve as the biological infrastructure of a post-plastic economy, a world in which the materials of everyday life are grown rather than manufactured, composted rather than buried, and in which the distinction between a building material and a living organism is not as clear as it once seemed.
This is not science fiction. The science exists, the engineering exists, the companies exist. The question is whether we can move fast enough and scale these approaches aggressively enough to make a meaningful dent in the plastic catastrophe before it becomes irreversible.
Let's bring this a little closer to home, because if you've made it this far, you're probably not just casually interested in mushrooms. You're the kind of person who has noticed the mycelium colonizing a log in your backyard and felt a deep and perhaps inexplicable satisfaction. You're the kind of person who has eaten a mushroom you identified yourself and felt, in that moment, connected to something ancient and real. You're the kind of person who gets it in a way that many people don't when somebody talks about fungi as more than just organisms, as a principle of biological organization, a mode of being in the world that is radically different from anything we experience as animals. And that means you have a role to play in what happens next.
At the most basic level, you can support the science. Citizen science programs in mycology are underfunded and understaffed, but they are enormously valuable. Projects like iNaturalist's fungal observation databases help researchers understand the distribution and diversity of fungal species, information that is essential for identifying which organisms might harbor plastic degrading capabilities and where they are most abundant. If you're out in the field anyway, recording and sharing your observations costs you nothing and contributes directly to the scientific knowledge base. You can support the companies and research institutions doing this work, whether by buying their products, following their research, or simply talking about what they're doing. The mycelium materials industry is growing, but it is still fragile, and public awareness and support matter enormously for the investment decisions that will determine whether these approaches scale. Every time you choose a product packaged in mycelium composite over one packaged in polystyrene, you are casting a vote with real economic weight. You can experiment in your own right. Citizen mycologists and amateur cultivators have contributed to the scientific understanding of fungi in more ways than the academic establishment sometimes likes to admit. If you are growing oyster mushrooms, reishi, turkey tail, or any of the other species with known plastic degrading potential, you are sitting on an extraordinary experimental system. The observations you make about how your cultures respond to different substrates, what they colonize readily, what they resist, what unexpected materials they seem to degrade, are potentially scientifically interesting. The Fungi Foundation, the North American Mycological Association, and numerous academic mycology programs are actively interested in engaging with the amateur community. The door is more open than you might think.
You can educate people around you about what fungi actually are and what they are actually capable of. We are, as a culture, profoundly mycologically illiterate. The average person's model of fungi is limited to the mushrooms on their pizza and the mold in their bathroom, and the idea that these organisms represent an ancient, sophisticated, and genuinely powerful biological kingdom with profound implications for the future of our civilization has simply not penetrated popular consciousness. Every conversation you have about this, every time you explain to a curious friend or family member what mycoremediation is, or what the Wood Wide Web actually does, or why the fungi at Chernobyl are one of the most remarkable discoveries in modern biology, you are doing genuinely important cultural work. And perhaps most importantly, you can simply pay attention. Spend time in the forest, in the meadow, in the soil. Watch the mycelium. Follow the fungal stories as they emerge in the scientific literature and in the news. This field is moving fast, and the next several years are going to bring discoveries that will surprise even the most knowledgeable mycologists. The plastic-eating fungi we know about today are almost certainly a small and unrepresentative sample of what actually exists. The enzyme optimizations currently in progress are going to dramatically change the practical landscape of mycoremediation. The mycelium materials industry is going to bring products to market that challenge our fundamental assumptions about what buildings, vehicles, and consumer goods can be made from. This story is not finished. It is barely begun. And the people who understand fungi, who have taken the time to learn what these organisms actually are, who have built a relationship with them through cultivation or foraging or simple observation, are going to be in the best position to appreciate, contribute to, and benefit from what comes next. So let's come back to where we started. A student in Ecuador, a mushroom growing out of a piece of polyurethane foam. A discovery so improbable that it barely seemed possible, and yet so consistent with everything we know about the biology of fungi that in retrospect it was almost inevitable. How could a fungus learn to eat plastic? The same way fungi learned to eat lignin, which was once the most recalcitrant material in the biological world. The same way fungi learned to survive in the intensely radioactive environment of Chernobyl. The same way fungi learned to help plants colonize bare rock and create the soils in which all terrestrial life depends. The same way fungi over and over again throughout 1 billion years of continuous evolutionary refinement have found a way into every material, every environment, every niche, every seemingly impossible challenge the planet has presented them with. By having time, by having diversity, by having a networked, exploratory, information-integrating biological architecture that is, in its own way, one of the most sophisticated problem-solving systems in the history of life on Earth. How has nature begun to fight back against the plastic nightmare created by humanity? The same way it always does. Slowly, quietly, not with a dramatic reversal, but with the patient, incremental colonization of a new substrate by organisms that have nothing but time and capability on their side. In soils around landfills, in ocean surface films, in agricultural fields where plastic mulch has been used for decades, the plastic-eating fungi are already at work. Evolution selected for them the moment the first significant plastic contamination appeared in their environment. We just weren't looking. And what if I told you this fungus could prevent a future catastrophe? I already have, and the evidence suggests that it can. Not alone, not quickly enough if we continue producing plastic at current rates,
not without our active assistance in understanding, optimizing, and deploying these biological capabilities at the scale the problem demands, but combined with a genuine effort to reduce plastic production, to deploy fungal and enzymatic bioremediation at industrial scale, to replace plastic with fungal materials wherever possible, to support the science and the companies doing this essential work, yes, the fungi could help us find a way out. They've been finding ways out of impossible situations for 1 billion years. I wouldn't bet against them now. The next time you step into the forest, look down at the soil beneath your feet. Somewhere in there, a few centimeters below the surface, a hyphal tip is probing through the darkness, sampling the chemistry around it, integrating information from across a network that may span hectares, deciding, in whatever sense that word applies to a network of interconnected fungal threads, what to do next. In that decision, made in the dark and the damp and the silent complexity of the forest floor, is an intelligence, a biological sophistication that has been refined since before animals existed, before plants existed, before the continents settled into their current positions. We have an ally down there, one we've barely begun to understand, one that is right now teaching itself to eat the mess we've made. Pay attention to the fungi, share what you learn, get your hands in the soil, and spread the word, because the most powerful thing a mushroom enthusiast can do right now is help the rest of the world understand what fungi actually are and what they might just be capable of doing for all of us. Thank you so much for watching. If this video sparked something in you, a curiosity, a question, an urge to look more closely at the next mushroom you encounter, then please like and subscribe so you don't miss what's coming next. We're going to keep diving deep into the fungal world and the science that's revealing just how central these incredible organisms are to the future of life on Earth. Drop a comment below. I want to know, which plastic-eating fungus surprised you most? And if you've been doing any of your own mycoremediation experiments, share them. This community is one of the best resources we have. We'll see you in the next one. Sources and further reading available in the description. The science of fungal plastic degradation is moving fast. Check back regularly for updates.